Hi everybody, I'm Matt Warman. I work at Boston Children's. I'm, I guess, a core co-director of the RNA sequencing core. And Catherine today is predominantly going to talk about bulk RNA sequencing, but we can offer the same sort of advice and consultation for single cell experiments. And I hope that at some point during the P30, we'll be able to offer spatial transcriptomics, which is sequencing histologic specimens on glass slides. I had a postdoc that was working on it, but she left to go take a clinical job. I have no one working on it, but Peter Nigrovic, who's coming to Children's to direct the immunology program from the Brigham, is also working on it. So if he gets to it before us, I'd say we use his core, or maybe we'll get there at the same time. And 10x Genomics is also working on it. They bought the company that does spatial transcriptomics. But last I spoke with my contact there, former grad student of mine, they had not yet gotten it working for cartilage or bone. So I think when they do, I'll probably hear earlier than most. And I'll let you know about that. So today, what Catherine's going to do is sort of introduce to you, the types of services that we can offer in the core. And the one other thing that I want to point out is Diana Corleone gave a really nice talk on RNA sequencing the other day. And she's obviously done a lot of it and thought about it, as have several other people probably on this Zoom conference call. So I would love if we could serve as a sort of gathering place for protocols that people like, for technical advice and for tips that people have sort of learned along the way, that we can post on the core website, and people can then download protocols that might be specific to their topics of interest. And Catherine will get to that towards the end. So Catherine's been in my lab, it's been about a year, a bit less. She completed 2 masters, her latest one at Boston College. And she's a bench scientist, but she's also quite good computationally. And so she's going to now introduce the RNA sequencing core. So Catherine, shall I turn it over to you?
Sure.
And oh, the other thing is the talk is not as long unless it's extended. So you should feel free to interrupt and ask questions along the way.
Thank you, Matthew, for that introduction. And thank you all for coming today to this introductory talk on the Center for Skeletal Research, RNA-Seq core, which is hosted at Boston Children's Hospital. My name is Catherine May, and as Matt said, I have 2 master's degrees in molecular biology and bioinformatics. I joined the Boston Children's Hospital in January and have since taken 5 courses with the Harvard Chan Bioinformatic Core, as well as a computational biology course with the Broad Institute. And I've been developing analytical resources for the RNA-Seq core. So today my talk is geared towards a general audience, and for some of you this may be a review on the basic concepts of RNA-Seq, but we're going to tell you what we can and can't offer you with the Bone RNA-Seq core and a few of the things that we've developed. So today I'm going to discuss the pipeline that we worked on. It's based largely on the Harvard Chan Bioinformatic Core pipeline, which is open source and available on their website for free. I've also helped create a bioinformatics working group with the help of April Kraft, and that involves several labs in our department that work on many different kinds of skeletal data sets and many different model organisms. So working with this diversity of data has helped us to broaden our understanding of frequently encountered issues, which on the surface may appear basic, yet they're persistent issues we see in next generation sequencing analyses. So during our working group meetings, we screen share and collaboratively problem solve on Zoom for both bulk and single cell RNA-seq analyses. We deliberate heavily on the best standards and most up-to-date methods in the field, and thus we can offer you valuable expertise and advice to the CSR community. So shown here is going to be our main pipeline that we work with. But I'm going to start off with a quick refresher on RNA-Seq. Working with skeletal tissues, either bone or cartilage, presents unique challenges such as isolating RNA from hard tissues quickly enough to prevent RNA degradation and changes in transcriptional profiles. Variation in tissue pulverization and collagenase treatments have been shown to affect differentially expressed genes. Thus, minimizing the timing of extraction is critical, as is standardizing the timing to prevent sample bias. Although we commonly use polyase selection to reduce ribosomal RNA, we still see quite a bit of rRNA present in our data sets. So I have written code to additionally remove rRNA in post-processing such that it does not skew our analyses. One important thing to consider is that only 10% of transcripts are captured with RNA-Seq. And of that small percent, 98% are going to be ribosomal RNA. So it's very important to consider that lowly expressed genes may be difficult to detect and that your quantification will be inherently biased. In addition, it's been shown that RNA will actually be retained on plastic tips and tubes, thus severely depleting your mRNA. So it's important to use low retention tips and plastics. Also, not all kits are compatible with DNases. So if you're going to use DNase to remove contamination, just keep that in mind that you want to be compatible with your kit. So what is the goal of RNA-Seq analysis? We want to detect differentially expressed genes to evaluate whether a knockout, a mutant, or a treatment group has a causal impact on gene regulation in disease. However, during RNA-Seq, there are many steps at which factors can create quality issues. So as a quick review, you're going to first isolate your RNA, reverse transcribe, do cDNA synthesis, and you're going to add barcodes, adapters, and indexes. The library is then going to bind to a flow cell adapters and undergo bridge amplification. And in sequencing by synthesis, when each nucleotide is added, it has a unique fluorophore. And these, basically your raw data is going to be actually pictures taken of the flow cell of each of these nucleotides. So these raw image files, or BCL files, are then translated into FASTQ files, which contain reads with the sequences right here. And these sequencing reads can be aligned to a reference genome or a transcriptome. So when we deal with this data, we look for things such as the quality, or how confident are we that that nucleotide is really an A and not a T. We look for things such as coverage, which is how much of your region of interest is covered by a specific read, and also depth, how many redundant reads are aligned to any particular region of interest. So I think most people's intuition is that it's better to have more depth when you order your libraries. However, as emphasized by the Harvard-Chan Bioinformatics Core, it's actually generally better to have more replicates than greater depth. If you look at the graph here on the left, the more replicates you have, the more number of differentially expressed genes you can have. And you see huge gains actually from having 3 replicates to 4 or 5, for instance. So not only that, but your power to observe differentially expressed genes almost rises exponentially if you have 4 or 5 replicates. So having more replicates is very important for reproducibility, as there's often variability in these differential gene expression analyses. Another issue we frequently encounter in our bioinformatics working group is batch effects. So consider the following question. Is it better to have one batch with all 3 conditions and all 3 replicates together, or 3 batches with a representative replicate in each batch? Now, in reality, it can be difficult to have all your samples in one batch because breeding varies, culture length of tissue or cell culture varies, and libraries may have to be split along lanes when you're sequencing. And as we all know, biology is messy. So ideally, you'd like to freeze your RNA and conduct your library preparation on the same day, but this can also be problematic. Consider the classic example of precision versus accuracy. With precision in your samples, all of your samples are close to each other. And with accurate samples, your samples are close to the true biological center. yet this is often not knowable. However, if you only have one batch, it is not possible to know what your technical variation is, were you to repeat the experiment multiple times. So this can make it difficult to compare between studies and between labs. So it may be strategic and necessary to plan for batches, particularly depending on the sequencing platform you're using. For example, NextSeq can produce 40 million reads, so multiple sequencing batches may be necessary. But the NovaSeq produces 2 billion reads and may only require one sequencing batch. So how do you choose to divvy up your samples on lanes, and what platform you use can be critical? So we record all this information about batch in something called metadata, and that is going to be all the data relevant to your experimental details and not necessarily your primary data set, which is the RNA count data. In the previous example, I showed you 3 different conditions with 3 different replicates, but I only explained it in the context of having one batch. But what is a batch and how do we define it? There are many types of batches, including the animal from which tissues or cells was isolated, cell or tissue culture isolation, RNA extraction, library kit used, and the sequencing batch. So it would probably be cost prohibitive to have 15 replicates per sample with 3 different conditions. However, by strategically splitting your samples up to generate batches that represent all your confounding variables, you have the possibility of regressing these factors out, improving the accuracy of your data, and increasing the reproducibility of your study. So batch effects are something we quite often discuss in our bioinformatics working group, and we are happy to offer advice and expertise to the CSR community. If you're contemplating doing RNA-Seq experiments, please feel free to give us a call. We can give advice to you on stopping points and batching, and Matt is happy to have those conversations if you need advice.
Yeah, I'm just going to butt in for a second here. And we and many other people have learned the hard way that you think RNA is RNA. And once you have RNA, you're going to just get it sequenced, just like DNA is DNA. But there's so many confounders, as Catherine talked about. What the temperature of the incubator was when you harvested your cells, whether you killed your animal in the morning or in the afternoon, whether you made your library all at once or you made libraries on different days. So, so even before you contemplate beginning an RNA sequencing experiment, it's worth reaching out to the core and explaining your experimental design so we can get you to think about things that you may not have considered.
Thank you, Matt. Next, we will discuss quality control for RNA extraction. So how do you evaluate if your RNA extraction is good? We use a Bioanalyzer, and these services typically cost around $40 a sample, and many facilities offer these services. In the graph on the left-hand side at the top, you'll see a spectrum of RNA. The ribosomal 18S and 28S are the bands here. And a sample that's partially degraded, what you'll see is a shift towards shorter fragments, a decreasing ratio of 18 to 28S. You'll see peaks below the bands and an overall decrease in RNA signal. The RIN, or the RNA integrity number, indicates the quality of your RNA and whether or not it's degraded. In general, an RIN of 7 or more is good. However, even if you have excellent RINs in your sample at very high levels, if your samples have varying RINs, we have found that they can actually cluster on a principal component analysis by RIN alone. So to combat this, we've been including RIN data in our metadata sheet, and that allows us to regress this out if needed. Next, we'll talk about library preparation. You can also use a Bioanalyzer not only to quantify the quality of your RNA, but also your library. For library prep, you can do it yourself, or you can send it out to a sequencing facility at about $200 per library or less. And one benefit of outsourcing your library prep is that at high throughput facilities that use liquid robotics, you have a much higher reliability. And they also often do RNA spike-ins so that you can absolutely quantify RNA. One con though is that since they're through HiPut, you usually have a minimum of 8 or 16 libraries, and you may have to wait until the entire flow cell is occupied before they run it. So you may have to wait. But how do you know your RNA library is good?
Oh, so let me just expound upon that just a little bit. So April Craft, and I think many of us have made our own libraries. And one advantage of making your own library is if you have a teeny tiny amount of RNA that you're starting with, one cartilage cap from a mouse's femoral head, and you have picograms or nanograms of RNA, you can order these very low input library preparation kits and make them yourself. Whereas if you send them to a company, very few companies are going to be willing to make a library for you if you don't send them a decent amount of total RNA to begin with. However, if you have a decent amount of total RNA, and as Catherine said, you're going to want to do a fair number of replicates, because the more replicates you do, biologic replicates, the better power you have to find significantly expressed genes. Then sending 8 samples or 16 samples at a time is not out of the question. And the companies can offer to make libraries anywhere from like $100 to $175 a library. I think our costs, and maybe April can chime in. April, have you costed out your library cost? I think last time we did it, it was about $70 to $100 a library material cost just to make them ourselves.
Yeah, it ends up being cheaper almost to send it out.
Yeah, and then as Catherine was saying, the robotics, it's not that they're on an analyzer at the same time. They're done in 96 well or 16 well plate format with robotics machines that always pipe in the same amount, all at the same time. So the quality of library prep is probably more consistent if you do all your samples through a company. And it doesn't have to be through a company like GeneWiz. It could be at the Broad Institute. It could be at the Biopolymers facility. I'm sure Mass General has a facility that does library prep as well. But critical is, even if you're going to store your RNAs because you're extracting them at different times, have the libraries made at the same time if at all possible. And if not possible, make sure that you always have experimental and control samples being turned into libraries simultaneously. And again, Catherine will give you an example of that a little bit later in her talk.
Absolutely. So once you bioanalyze your library, how do you know that your data are good? You want to look for a normal distribution centered around the peak of your expected fragment size. And you want to make sure that this is in line with the kit and your sequencing facility. So as Matt mentioned, many sequencing facilities offer bioanalyzer services about $40 a sample, as well as qPCR services. We do have a bioanalyzer available at Boston Children's Hospital, which you can use if you purchase your own reagents. If you're interested in using our facilities, you can contact our lab manager, Steve Hahn, and he can help facilitate that for you. Okay. So next we'll talk about RNA sequencing. There are, of course, many options for sequencing that might be right for your purposes. However, we do recommend using paired end sequencing. Although single end sequencing is much cheaper, it's difficult, if not impossible, to identify PCR duplicates without having a paired end library. Your alignments will also be higher quality than with single end reads alone, and have a higher potential to capture splice sites and isoforms if that's what you're interested in. So the trade-off with smaller reads is one of price and quality. We have experienced lower alignment with 35 base pair read data sets. So I personally would recommend at least 150 base pair reads if possible. This also allows you more flexibility should you need to perform trimming on your reads if you have quality issues. So how you pool libraries among lanes is also something that requires careful consideration, as each of the lanes on this flow cell can almost be thought of as a technical replicate. You want to make sure that each of these lanes contains all of your treatment types. So you don't want one lane to have all wild type and one to have all knockout. While prices vary by institution, if you contact us, we can provide you advice on sequencing providers such as GeneWiz, the Bro, the Bauer Core, and Biopolymers. It can be difficult to get pricing, and in fact, I went on the websites trying to have comparable price points, and it's notoriously difficult to pin down exactly what things cost. But if you have questions, you can contact us and we can give you our best advice. So next I'll talk about quality control for sequencing. The first thing we do when we get these FASTQ files back is to evaluate the quality of the data set. So how do you know your data are good? We look at things such as read length. Do you get the length that the company has promised you? Do you get the quantity that they promised you? How is the read quality? Is there a bias, 5' or 3' bias? What is your GC content like and how does it compare among samples? What are your duplication levels like? And how is your library complexity? Do you have a low complexity library that indicates problems during the PCR amplification stage? These are things that we look for commonly. And also, what % of your reads map uniquely to a reference genome or transcriptome? So FastQC is a program we use.
So you may want to go back and explain that bell curve slide.
Oh, sorry about that. Okay, so this is off of one of the FastQC reports, with blue being the ideal theoretical distribution of mean GC content. And then of all the reads in the library, the red curve is what shows the actual GC count per read. Now, if you've got a skewed distribution that's not following the theoretical distribution, there can be many problems with your library, including contamination from other species, ribosomal RNA, 3' or exonic bias. There's a lot of different things that can happen, but what we can do is look at your mean GC content and indicate if there are issues. Thank you, Matt. Do you have anything else to add on that?
No, it's just, as you mentioned, it's one of the quality controls. What do you expect to get from good sequencing reaction, and do you actually get it? So the transcriptome generally is about, what, 49% G or C containing? And if your transcriptome from your data set is only 29% G or C containing, then you probably had a PCR hiccup somewhere along the way. And if it's like 80% G or C, it's probably got way more ribosomal contamination than you thought. So these are little things that we can help you to understand if your data are not yielding what you thought they would be, why they're not. One of many. Catherine's going to show you others.
Okay, so in the FastQC report, we provide to our CSR clients. We covered many basic statistics here, including things like, yes, what your GC content is. Do you have overrepresented sequences that are a result of PCR duplication? And if adapters are present, we can also trim those as well. So in this graph below, this shows the read position and read length. So from this, I can infer this is a 75 base pair library. And the colors indicate the quality, with green being good and red being bad. And roughly the quality score, or what's called like Q30, for instance, here, means that you have 99.9% confidence in the accuracy, whereas Q20 is 99% and Q10 is 90%. So you definitely don't want to be in this red zone. Now this data set has excellent quality. You can see it's all in the green zone up here near 38, with only minor variations at the 3' end. In addition, this tells you the total sequences here. So here we have about 5 million reads, and this is only for one end in a paired end library, so it'd be about 10 million reads. And it shows you GC content as well. So here's an example of a problematic data set. The read quality is good all the way up to about 35 base pairs, after which we see a precipitous drop-off. So these are definitely things we look for immediately before we even do the DE-seq analysis, the RNA-Seq analysis. However, this data set does not necessarily need to be discarded. We also provide multi-QC reports, which allow for quality comparisons between your samples. So we look at things like how many million sequences did you get? Is it what the company promised you when you ordered it? We also look at percent duplicates. And I'd like to point out here that there's a difference between natural duplicates resulting from high gene expression and PCR or optical duplicates. So I use another program called Picard to distinguish between these two types of duplicates. And as we said, we also look at GC content. You don't want these levels to be too disparate between different samples in your data set.
So I'm going to just butt in there just to make one other point. So a really good library that's very complex, where you had a good amount of starting material, might have a PCR duplicate rate of 10 to 15%. And the reason why there's a PCR duplicate rate is when you actually make your RNA into a cDNA and fragment it and add adapters so that you can sequence them, in order to get enough DNA to put on the sequencer, you have to do a few rounds of PCR. So you're going to get duplicates because you have to do a few rounds of PCR. If you have a very small amount of starting material, let's say a library from one mouse's cartilage from the femoral head, compared to a huge plate of cells from tissue culture, you're going to have to do more PCR steps to make enough material to put on the sequencer from the femoral head cartilage compared to from a plate of cells. So the duplicate rate is going to go up. And it's not a problem to have a high duplicate rate necessarily, but it does influence how you're going to interpret some of the data. So again, by looking at PCR duplicate rates and the libraries you try to make and what the starting material is, we can tell you whether there's going to be technical artifacts introduced during library preparation that might obscure some of the biological truths that you're looking for.
Mm-hmm. Precisely, and there have been some experiments where people will tag, basically tag individual molecules, do the PCR reactions, and then see how much this duplication's interfering with it. So, you know, there's a lot of new software being written to try to deal with the problem of duplication, to identify PCR duplicates versus true biological duplicates. So we also provide read alignment files, are called BAM files, which can be opened up and interactively viewed in IGV or Integrated Genome Browser. It's relatively easy, even if you have no bioinformatics experience. So in this alignment, we see the FGFR3 gene. We see a 5' tail here, end here, and a 3' tail over here. And we see a histogram showing the abundance at any given exon. So from this, we can investigate things like 3' bias, which may have arisen during cDNA synthesis. And we can see that there's clearly bias here. The 5' end is way underrepresented where the 3' end is overrepresented. And in genes with longer transcripts, the polymerase may fail to reach the 5' end, creating this bias. So this is a qualitative way of looking at bias, but how do we quantify bias? So we also provide quality map reports, it's another program, which can quantify things like exonic and intronic representation, as well as 5' and 3' bias. Now, a perfectly unbiased transcript would have a ratio of 1, and that's a ratio of coverage, say at the leftmost base pair, to the mean coverage. So here we see that 3' is overrepresented and the 5' is underrepresented. So for certain types of analyses, if you're interested in RNA-Seq variant analyses or SMP analyses, this is important information to have. So we can not only identify quality issues, but we can perform quality trimming. I use a program called Trim-O-Matic, and we can trim either by quality score of total read or the position in read. So in this case of this poor data set, we can truncate everything after 35 base pairs, and you can still use this RNA-Seq data. Now, for the issue of what to do with PCR duplicates, as I said before, it is a complicated issue. The industry standard is currently evolving, but there are new publications that show if you you can reduce false positives by up to 8% if you remove duplicates. And ideally, these PCR duplicates would be the exact same length in the exact same sequence. But in practice, it's really difficult to differentiate these between biological duplicates. So while we don't currently offer advice on whether or not to discard duplicates, we do offer Picard or FAST-P reports to give you that information so you can make a more informed decision and decide if you have a low complexity library. So next we'll talk about read count quantification, which is done in a program called R or RStudio. Our pipeline primarily uses Callisto, which is a pseudo alignment program that's very fast and very accurate for generating read counts. And it uses a reference transcriptome index. We also run another program called STAR, which is a splice-aware program that will align your reads to a reference genome. It is more computationally heavy and takes longer to do, but I typically like running both of them and comparing the results to make sure there's no major discrepancies. So once we get read counts, the first overview of the data we do is to look at highly expressed genes, which is normalized in TPM or transcripts per kilobase million. Once we generate normalized read counts, we can conduct a principal component analysis across the libraries and look for any potential issues such as outliers. So I have written some code to quantify sample outliers, and we also frequently use a leave one out approach, repeat the analyses, and check that our results are robust. In the past, FPKM and RPKM were popular methods of normalization, but they've kind of fallen out of vogue as newer methods have emerged. FPKM and RPKM are not ideal for comparing between samples because the sum of normalized reads in each sample may differ, so you can't quite compare them. In contrast, TPM, or transcripts per kilobase million, they're normalized by dividing read counts by the length of the gene and divided using a per million scaling factors. Since the sum of TPMs in each sample are the same, you can kind of compare the proportions of reads that map to each gene between samples. However, it's only DE-Seq2, which is what we primarily use, or EDGER that you can compare between samples and conduct differential gene expression. So we use DE-Seq2, and they use a normalization method called medians of ratio, and that's going to account for read depth and read composition. And EDGER uses trimmed mean of M values to normalize, and that actually allows you also to compare within sample. However, the documentation's a little bit less developed, that it's not as straightforward to adapt scripts to work with, but we do have some EDGER scripts in our department that we can also do if you're interested in comparing read counts within a sample. So one of the other first things we look for in samples, particularly isolated from bone, is tissue contamination. So in this sample, we see evidence of blood contamination, as well as muscle contamination.
Catherine, you have to explain the Excel spreadsheet.
Okay, sorry, here are highly expressed genes. We have read counts for different samples in TPM, transcripts per kilobase million. We have the gene symbol and the ensemble gene ID here. And ACTA1 is our most highly expressed gene. So this isn't differentially expressed genes, it's simply what is the most highly expressed gene. And what we see here is a muscle gene. We see 2 muscle genes here in the list and 3 blood cell genes as well. So this can be problematic since we don't really care about blood or muscle tissue, and we're primarily interested in bone tissue. Matt, did you want to add something?
So this is from Or Iturk's original bone RNA-Seq paper from 2003. And he was very good at getting the bones out, spinning out the marrow, and cleaning off the periosteum. But even being really good at it, anytime you had some contaminating muscle, there's so much alpha-actinin 1, and there's so much troponin C that it just swamps out a lot of the bone-specific transcripts. Same thing for if there's blood contamination. Red blood cells, if they're buried in the small veins coursing through cortical bone of a mouse that you can't get out, well, red blood cells are just big bags of hemoglobin with lots of hemoglobin mRNA in them. So by looking at bone sequencing or any tissue that you think you're sequencing, you see by just looking at abundance of transcripts that there's contamination. And there are ways of dealing with this contamination and honing in on the transcripts that you're interested in. So at least the good news here among the first, 15 most highly expressed transcripts in wild type bone from animal S114020 is the alpha-2 chain of type 1 collagen. And the alpha-1 chain was actually not that far behind. So there really are a lot of bone-specific genes here. But by just looking at the abundance of transcripts in your samples, we can help you sort of think about contamination, degradation, signs of cell stress that you might not have thought have been occurring as you harvested, say, tissue culture cells or ex vivo cultured cells before making your libraries. So this, just the abundance of transcripts in an individual library can be very informative. And here there are 3 columns of 3 different bone RNA libraries that Orr made. And you could see, for example, that the hemoglobin BS and HbA2 are reasonably similar levels, whereas the muscle contamination of the far right column is about half that of the first column, 19,000 for ACTA1 versus 39,000 in the middle column, 9,000 for troponin, 92,000 for troponin 2, T and C2 at the bottom versus 224,000. So clearly there's twice as much muscle contamination in one sample as in the other. We can help in the analyses to control for that. So if you bring us data, Catherine can really help you analyze your data to get meaningful results from it. Next slide.
Yeah, right. And since it's such a persistent problem with trying to isolate bone and skeletal tissue, we really should think about a way of how to normalize it, normalize for tissue contamination. And so that's something that I put on my list of things to work on. So yeah, we want to evaluate how similar libraries are to each other. And not only can we just look at a list like this, we can also do a principal component analysis. So a PCA, everyone should be familiar, but in case you're not, it's a statistical technique that takes very high dimensional data, i.e. 10,000 genes, and it calculates the influence of those genes on variation in the data. And it reduces the data and allows us to plot it in a 2D manner such that we can compare similarities and variance between samples. So in this example from April Craft's lab, H9 embryonic stem cells were differentiated either into growth plate cartilage or articular chondrocytes using BMP4 or TGF-beta3. And each circle represents a sample. Now, we would expect to see that PC1, our greatest explainer of variance, is treatment group. That is, cells that were subjected to either BMP or TGF-beta. However, what we see is that PC2 is what divides these groups, and PC1 is an unknown cause of variance. But thankfully, April is meticulous about recording metadata for her experiments, and this allows us to probe into this issue deeper. So I've talked a little bit about metadata, but I'm going to explain it in a little bit more detail. It is all the factors that go into an experiment and influence your primary data, which is the RNA-Seq data. We record everything. It's critical to do so, including genotype, whether or not it was subjected to collagenase, for instance, the culture duration in weeks, when tissues were collected, the library prep, sequencing, etc. And the CSV file with this metadata is read into RStudio along with the bulk RNA-Seq counts, and it can be incorporated into the design formula. A design formula is what DE-Seq2 uses to model RNA-Seq counts. So with the formula below, counts explained by batch plus treatment, it regresses out the effect of batch and allows us to focus only on treatment. So this improves the quality of our analysis. So here we see about 80% of the variance is unknown, but if we probe deeper, we can see when we color samples not by treatment type but by batch, that PC1 is being divided largely by batch. Had April not properly divided her batches, making sure each one had both BMP4 and TGF-beta3, there would be no way to tell what is driving the variance in PC1. But because she's diligent about including all treatment groups and batches, we can identify batch as a major confounder and regress it out accordingly. And this all seems very elementary to say, but experimental design is critical at every step of the way. However, we still don't quite know what variable in this batch is a main driver of variation, because culture duration, collection date, library prep batch, and sequencing batch were all done concurrently and thus are collinear. So for future edification, one might want to intentionally divide batches by each of these variables if you want to pinpoint what variables are the major sources of experimental variation. And this can be really useful if you want to replicate future experiments and you want to know which ones are the most variable factors that you work with. While we don't know what variable is contributing to the batch variation, we can still regress it out. And this improves the accuracy of our differentially expressed genes and allows us to identify transcriptional changes that are only attributed to BMP or TGF-beta. So the question is, if you ran the same experiment again, how similar would your results be? In other words, how robust is your data? So I'd just like to reiterate again that the more replicates you have, the more differentially expressed genes you can detect, and the greater your power to detect them. And furthermore, if you only had one batch, you can tell how precise your data is or how close your samples cluster, but you wouldn't know the technical variation necessarily, and you wouldn't know how accurate your results are. So it's my personal preference, but I want to be able to know and quantify what that technical variation is, and I want to be able to build it into my model so that I can regress it out properly. So if you have the extra money, I definitely recommend at least doing 4 or 5 replicates if you can. I know it's cost prohibitive, but the quality of your data is going to be much greater. You're going to be able to reproduce your data much more reliably. So we can also use PCA to identify sample outliers. In the graph below, I've covered each dot by sample name and identified TGF-13 replicate 1 as a potential outlier. And even though all the samples were processed in the same way on the same day, this particular sample does not cluster with its other replicates. So how do we make decisions on whether an outlier needs to be kept or removed without running into the danger of cherry picking our data? As a rule of thumb, there are several less biased ways of evaluating outliers. For instance, you can use a z-score. So you can find the replicate average for your samples, calculate the z-score, and check if that sample is more than 3 standard deviations away from the sample mean. I've also written code to pull up the top DEGs contributing to each principal component for that sample to see what top genes are driving that PC and whether or not they're biologically relevant.
Catherine, you want to say that again and define DEG?
Differentially expressed genes. Yeah, so each PC behind the scenes in our studio, there will be a list of genes that are contributing to that. And you could think about this as influencing, pulling these samples in one direction or another on the PCA. And so if you have one gene that's really strong influence, it can drag kind of the sample one way or the other. And each of these genes are kind of weighted, and basically you can see which ones are really responsible for the location of them on the PCA. So I have code to pull those top genes that are contributing to PC1 or PC2, et cetera. Yeah. So you can also do a leave one out approach. So you can remove this outlier, redo the PCA, and see if you still get the same results. But April's intuition is it's better probably to do more replicates than to actually remove data points and run the risk of biasing your data set. So lastly, I'm going to cover differential expression. We can help you also with that, perform your DEG analysis. Our pipeline includes optimizing the data set to account for the natural distribution of RNA counts. So RNA counts have an interesting distribution. They have inherently high variance at low counts and low variance at very highly expressed genes. So DE-Seq employs something called shrinkage, and that's a method that's shown to improve the accuracy of modeling. So we do that. And we've spent a lot of time writing and updating statistical analyses to check for things like contamination, cell stress, mitochondrial DNA, cell cycle, ribosomal RNA. So in this volcano that uses Orr's osteocalcin data, he compares osteocalcin knockout mice to wild type mice. And we can see that out of 12,000 genes, there were hardly any changes in transcription, and only 13 were significant. And interestingly enough, 13 of those genes were located on the same chromosome. Matt, did you have anything to add to this?
Not 13. I think there were 13 genes that reached significance, and I think 8 of them were on the same chromosome as the osteocalcin gene BGLAP that he targeted, making you think that removing a chunk of DNA that included the 2 BGLAP genes somehow influenced expression of other genes on the same chromosome. Several of those genes were a few million base pairs, or within a few million base pairs of the gene he knocked out. So it just food for thought. But again, the idea, this is a very simple plot, and you might want to explain what the X and Y axes are here.
Oh, sorry. Okay, so the log two-fold change. You have the log two-fold change, which is kind of the magnitude of how much gene expression is changing, and the p-adjusted log, negative log 10 value, which shows us how significant it is. And here's BGLAP, obviously was knocked out, so it's going to be one of the most highly differentially expressed genes in this study. So along with our analyses, we can provide publication quality graphs like the heat map shown below, and this is from April's lab. And in this example, we have 3 libraries per treatment group shown. And the heat map shows the normalized counts and the relative expression of genes between samples. So here we can easily compare samples and spot an outlier. For instance, one of these BMP4 samples highly expresses ALPL, unlike its other replicates. So here's when you might want to consider doing a leave one out approach. In addition to bulk RNA-Seq analysis, we also do quite a bit of single cell analyses, which can be notoriously difficult to work with. My script includes all of the following listed here, and our bioinformatics working group spends quite a bit of time evaluating each one of these steps and discussing best practices for quality control. And the RNA-Seq core can help you work with these single cell analyses and interpret your results. Our single cell pipeline here, it's a little bit more complex. You have cell isolation and gem information, something called integration and clustering, which is done with Surat inside of our studio. If you have any questions about that, feel free to ask. So what can we help you with at the CSR RNA-Seq core? We can offer you advice on how to best design your experiments and save you from very costly mistakes. We can offer access to our facilities for skeletal tissue pulverization or our bioanalyzer services. Our lab manager, Steve Hahn, can help you with that. And my main focus is data analysis, and I spend much of my time now coding and doing statistical analyses in our studio. If you'd like to utilize our data analysis services, we provide in-depth quality control. We provide publication quality figures, raw data, and Excel spreadsheets. And in addition to that, I think it's very important also to include our studio scripts. Now, this is going to be the raw code that shows each statistical step in the pipeline. And what I do is I produce an HTML file that you can look at in a web browser. It's got an interactive menu and it's very easy to browse through. You can kind of see the settings that were run. And if you have any students or lab members that want to learn these analyses, it's very easy to just start copying and pasting and learning how these things actually work. In addition to that, we can help you interpret your results. And I've also created a way to make transgenic references so that you can quantify your fluorescent genes of interest. So if you do flow cytometry and you've got GFP expression, I can quantify those read counts as well. And as I mentioned earlier, we do single-cell RNA-Seq as well. So for our data analysis services, the first hour is free if you want a consultation. And then after that, we charge about $100 an hour. And we roughly have 3 different packages. It really depends on how many samples you have and how deep you want your analysis to be. So pricing will vary. For use of our tissue pulverizer, we charge about $100 an hour. And single cell obviously is going to take a lot longer. It can be up to 10 hours or more depending on how involved it is. We give you all these wonderful deliverables, methods documentation, so you can reproduce the same sorts of analyses. For more extended projects, Matt is available for collaboration, so feel free to contact him if you are interested in a collaboration on analysis. And I'm currently building a website for the RNA-Seq core. Right now it's temporarily hosted on WordPress, but we're probably going to migrate it to the MGH or the BCH website. It still is a work in progress and we're adding new materials all the time. But right now I'd like to solicit the CSR community and ask for contributions or volunteers. If you have particular methods or protocols you'd like to volunteer, we would love to share them on our website. If you have particular expertise and you want to create a short PDF or a short video, we would love to welcome that. And we can list you on our website, and you can list that on your CV as community outreach. So right now I'm going to exit and briefly show you the website. So this is the website as is. If you go to data analysis services, there's 2 different things. We can tell you what we offer, and you can also access forums and different websites to order services. So if you go, what do we offer? It kind of tells you about the packages a little bit. It tells you kind of all the things I went through today, the reports, the quality reports that you receive, what kind of files you'll get. And here's an example of one of those HTML files I told you about. There's an interactive menu you can click easily and jump right to the volcano plot or the heat map or the violin plot. So it's very easy to browse. And these are great for sharing with collaborators. It's really easy to communicate using that. So we have that. And the other thing I'm really interested, like I said, is offering resources to the community for outreach. So under methods, we would like to add more protocols. If you do have protocols on the best way to extract or different aspects of it, we would love to add some of this as well. For our studio, this is a really steep learning curve for a lot of people. It can be very difficult to learn how to do these analyses. So what I've done is create PDFs, and here's an introductory to our studio PDF. It tells you how to get started, how to install things. So that's a very useful guide for people. And it also has the entire analysis here. And so let's say we want to look at volcano plots, for instance. What we've got here is the actual code used to produce those plots, as well as the actual images embedded in it. So people in your lab can actually learn how to do these analyses. And besides that, I also am working on creating videos in YouTube. So right now, we have only 3 videos up. But if you click on this, it actually will take you through an interactive, a way to interactively watch me do these analyses, and you can start figuring out how to actually do them yourself if you're interested. So the YouTube website is here. I only have 3 videos, but I'm going to keep working on this as well. And if any of you would like to contribute and you have a particular area of expertise, we would love to add more. This is a community effort. And I also have a GitHub where we're going to have the actual code written. These are studio files that you can download yourself and start playing around with and learning the analyses. So it is my goal to grow this resource. I mean, we are a community, and this is something that we should work on. Yeah. So with that, I'd like to acknowledge my boss and mentor, Matthew Warman, who's given me the opportunity to develop our data analysis services and educational outreach. April Kraft, who has been fantastically supportive and who constantly helps me improve my analyses. Steve Hahn is our lab manager, and he can help you with the pulverization or bioanalyzer services. And lastly, much of my pipeline is based on the Harvard Chan Bioinformatics Core. They have open source free courses, which you can teach yourself RNA-Seq analyses. And my personal contact information is here. Keep in mind, we're still updating the website and the YouTube, and hopefully it develops in the coming months here. But please contact me if you would like to volunteer at all. And with that, I thank you, and we'll take any questions you now have.
Catherine, that's great. Thank you for doing such a wonderful job.
Hi, Catherine. This is Paula. It was really wonderful. I just have a one quick question. When you were showing us, I think you call it the meta file, the file in which you have to record all the information of your data. Is there any specific format that you need to enter your information, or can you provide like a template, or any Excel spreadsheet will work? Because if I understand correctly, you can then use it to basically correct your data, your samples, right?
Right. Yeah, so it's a simple CSV file.
Right.
And you can just add columns, and these column headers is what you're going to call in our studio. So when I'm programming, I can say regress out experiment, regress out collection date, regress out library prep. So basically, as long as there's a column for it, you can regress it out.
But within the column, I can write whatever I want. You know, for example, the collection date has to be written that way, or I can write any date I want. That's what I'm trying to ask.
Probably do whatever you want. I mean, our studio can be a little finicky sometimes, but as long as this is something that can be identified, you can put quotations around it, and it knows that it's different. You can do it. Sometimes it's a little bit weird about numbers, but I've got in my script how to deal with that. Sometimes it likes ABC rather than 1, 2, 3, but it can be changed easily enough. Yeah.
Okay, great, wonderful. Thank you. This was really awesome.
Catherine, do you have a link to those open source courses?
Yes.
And do you have to be a Harvard person to take it?
Nope. And in fact, it is, well, I don't have it up right now, but no, it's right here. github.com/HBC training. They have a number of excellent courses. Sorry.
Yeah, there you go.
They have a number of excellent courses, including Intro to ChIP-seq, Intro to Single Cell, Differential Gene Expression. Their website is also here. Like I said, I took 5 courses with them, and it was really great in an interactive classroom, but you can totally self-teach. They have sample data sets, and they walk you through each example. They really explain the concepts very well too, which some of them can be deceptively tricky to learn. But yeah. Are there any other questions?
So let me just put in 1 or 2 concluding remarks then. And then someone else might want the last word. So RNA sequencing, like DNA sequencing, has come down in price. And lots of people are doing it, and lots of companies are offering it. But it can be a rabbit hole that you sometimes do not want to go down because you'll get lost. So we and others have chased the wrong gene due to technical artifact or some biologic phenomenon that we didn't understand. You can potentially get hundreds, if not thousands, of differentially expressed genes. And so deciding which ones to prioritize for downstream studies can be very challenging. And so as you're thinking about doing these experiments, it doesn't hurt, and we are happy to help reaching out to us so we can give you our 2 cents and hard-earned experience as you design experiments. And there are companies that will offer analyses for you as well. And if you just wanted to double check on it, you could come to us and we can have a look at it with you. And then I think our RNA-Seq working group, the group that you and April are doing, we've kept it somewhat closed because we discuss confidential data that we're not necessarily ready to share yet. So it's somewhat closed. But I think there are times in which we can certainly open it up, or we could try to find a way to acknowledge that our data are confidential, so it could be more of an educational environment for neophytes in the field. We'll have to think about how best to make that happen. Catherine, though, has been talking about offering some of these tutorials just independently of the working group that she and April and Jonah and Martha Murray's lab have all been, and Steve has been working on.
Yep, absolutely. I'm happy to organize a workshop if that could help. But also I think the YouTube is going to be very informative, as you can just watch me go through each section and actually analyze things and explain what each part of the code is doing. So that's my hope, is that you could have a student who knows nothing about RNA-Seq. They can come in, check out the Harvard Chan Bioinformatics core, download our scripts in a sample data set, watch my YouTube and be able to pick it up and teach themselves. That would be the ultimate goal.
Hi, it's Diana. Wonderful job, Catherine. The thing I found most difficult, and I don't do programming or anything, but I was given, the end result Excel spreadsheet to go through and analyze what genes have already been shown to be relevant to some version of the skeletal system. If it was limb development, if it was cartilage, if it was actual bone, so that you could pick out some of the really known ones. But it was hard because I would find something I thought was interesting and then I'd have to do all the background. So one of the things I would love, if there was a way of doing this, is set up databases so that we could scan our RNA-Seq data across databases and be able to pull out and say, these have all been identified. And so we can start narrowing things down. I can't tell you how many times I found something and had to really backtrack and say, well, was it ever shown in a limb? And that's the one thing I found really missing. And it's maybe me as being the novice, but I don't know if anybody else out there would be able to do it.
No, but it's not just you. I mean, resources for other fields and tissue types are very well developed. But if you look at skeletal tissue, it's very underrepresented. It would be great if I could write a script to include the most common skeletal, bone and tissue marker, bone and cartilage markers, and even in single cell to do automated cell identification. This is something I've thought about and I haven't quite had the time to do it. But it would be great to assemble some sort of database that you can do that. I mean, and furthermore, differentially expressed genes isn't really where our interest ends. We want to know more about functional pathways, what entire pathways are being upregulated. And to do that, I really feel like the community does need to come together and create some sort of resource based on what we already know, things published in the literature, things like cell differentiation. That's all stuff that needs to be developed. So if you have a way of organizing or trying to get people to do that, let me know,
But Catherine, you did have that in an earlier iteration of your talk, that there are like the gene ontology database and other databases out there that say, gee, the differentially expressed genes that you've identified many of them have been implicated in TGF-beta signaling or in Wnt signaling, or in inflammation, or in cartilage development. So those software are available, and you've used them. So that's a service that you can provide. And Diana, I'm sure you've used them as well, right? When you got your RNA-Seq differentially expressed genes back, I'm sure you went to the gene ontology database and stuff like that.
Mm-hmm.
Yeah, and I have code written for that.
We can help you.
Yeah, I have code written for gene ontology and CAG and all that. The question is, what do you do once you get that data? because we feel like, okay, you reach a point where you have these upregulated pathways and some make sense and some don't. And it's kind of a matter of, I guess, cherry picking them and then taking it to the lab and further probing it molecularly. But I still feel like we have a lot of computational work to make that into something that's meaningful. That's my only, yeah.
Yeah, but all those things are just hypothesis building mechanisms. So you're going to have to do that anyway.
Yeah.
So
For now, yeah.
actually, I have a question. Do you actually have any knowledge or any feedback into the effects of formalin fixation on the recovery of the RNA from samples, as well as what it might do to the sequencing?
Yeah, so formalin, I don't know specifically about formalin, but I know people are experimenting with methanol. Obviously during flow cytometry, there's some protocols where you fix cells. But because it takes so long to isolate RNA from bone and skeletal tissue, and you have to do all these collagenase treatments, I would love to see a protocol developed in which you can take tissue, dip it in methanol, freeze all of that RNA in time so the transcriptional profiles aren't changing as you're extracting, be able to maybe do flow cytometry and then RNA sequencing. There are some publications from different fields, not skeletal tissue, but maybe organ tissue, where they're doing this. And they claim that it's high quality. I think right now, I'm not sure that I don't know where we stand on that, especially for skeletal tissues. I think it's a little bit tricky and you would have to do the optimization. You'd have to do formalin fix versus fresh tissues, run the RNA-Seq, compare the two and really see how that is.
I'm sure people have done this because using samples from pathology, you're going to have to try to understand the effects of the fixation on the stability of your RNA.
Mm-hmm.
And like, laser capturing cells from sections and stuff like this.
Yeah.
I just wondered, it might be something that we could look at to try to have in our arsenal, that we know the artifacts that pop up and we know how you should best correct for them or be ready for them in either of those strategies. So, I mean, It may be that someone comes in like wanting to have RNA-Seq on laser capture, and, it would be good to know what that does to those samples.
I agree. Yeah.
The Broad has done that, and the Broad has done it. And part of the reason why they've done it is they're interested in somatic mutations, say in cancers. And so if they get a sequencing read that has a nucleic acid change from the reference sequence, they want to know, is that an oncogenic mutation or is that an artifact from formalin fixation? For most of our needs, where we're not necessarily going to be doing mutation detection, and we just are interested in transcript abundance and how it might change between tissues, those sequencing errors won't limit our ability to map the reads back to the reference genome. So we can still use it.
I wonder if that really is the case, though, because it may bias which transcripts can be more easily read than others. So is there a bias against longer transcripts as they may be degraded faster or something? I don't know what the scenario is.
Right.
But.
So what I would say is, if you want advice on how to look at formalin fixed tissue, we may never have done it before, but if you come to us, we would be happy to think about it, we being Catherine, and we would know to whom to reach out to sort of get those data sets that have already looked at it, whether it's people at the Broad, people at the Dana-Farber that have experience doing it. So if you know nothing about RNA sequencing other than you want to do it, and you want an intermediary to see if it's feasible to do and how you would control for the confounders that are in your experiments, we can be that intermediary. Well, we're after 5, and again, Catherine, that was just fantastic. Thank you so much.
Thank you.
For putting that together. And I hope people will reach out to us and start using our core now that our COVID-19 is, restrictions are being somewhat eased. Let's hope they continue to be eased. Bye, everybody.